So welcome, everyone. This is a meeting of Lower Hudson Group of Sierra Club. My name is Peggy Kurtz. We meet monthly online. These are our public meetings, mostly focusing on the climate crisis and climate actions on the local and state level. But for the last year or more, we've been also focusing on understanding the crisis of biodiversity and extinctions. It's a very serious crisis, and there's not nearly enough public attention to it. So what we're trying to do is to raise awareness, to understand it better ourselves, and then to begin to talk about what we can do. What are the actions that we can take, and we can urge public officials to take to what are the policies that would be more protective of biodiversity on which all life depends after all. So we started with a program on, well, I just want to mention before we go into our program that following our speaker, biologist Dr. Adriana Corrales, we will be speaking briefly about a lawsuit about a court decision, a court decision about New York State's failure to comply with some of the guidelines so far for the land break, the 2019 Climate Act. And then finally, at the end, we'll be talking about the protests in Rockland County and about the threats to the protests and what we can all do to protect freedom of speech in Rockland County. So we started this series on biodiversity and extinctions with a program on dark skies and about how light pollution impacts habitat. I want to mention that out of that program came a successful effort to get the Mario Cuomo Bridge to dim the lights overnight in bird migration season, which they are now doing. I first heard Dr. Corrales speak on NPR, and I was so fascinated that I wanted to share it with everyone. The first thing that really fascinated me was just the idea of how fundamental fungal networks are to nearly all plant life. So Dr. Adriana will speak with us, and then following her talk, we'll open it up for discussion. Adriana Corrales is a forest ecologist based in Colombia. Her research focuses on the ecology and taxonomy of tropical ectomycorrhizal associations and shifts in fungal community composition caused by biotic and abiotic factors. Her work has demonstrated key interactions between soil, biogeochemical cycling, forest function, and fungi in tropical montane forests. And she is on the board, I believe it is, of SPUN, which I don't have right in front of me. Let's see. It is the Society for the Protection of Underground Networks. Adriana, you should be able to unmute, and let me just make you a co-host so you can share your screen. Okay. You should be able to unmute and also to,
Can you hear me now?
yes.
Okay, great. Yeah, thank you so much for this invitation. I'm very happy to be here. I work for SPON, which is the Society for the Protection of Underground Networks, as the lead field research scientist and also the director of a granting program, the Underground Explorers Program. Let me hide this. I never know how to hide this bar.
It looks good.
Yes, but for me, I don't. Okay, let me see. Okay, now there we go. Now it looks good. Okay, so I'm gonna talk today about mycorrhizal fungi, which are the underground fungal networks that are invisible to our eyes, but basically could be the keystone species that are supporting life on Earth. So let's talk a little bit about what is a mycorrhizal fungi, right? Let's start from there because sometimes it's hard. A mycorrhizal association, a mycorrhizal fungi is a fungi that creates engage on this symbiotic interaction with the roots of plants. So these are fungi that live inside the roots of plants and form a mutualistic interaction, a mutually beneficial interaction with plants, when where fungi absorb nutrients from the soil and transfer those nutrients to plants in exchange of carbon. So carbon are easily acquired by plants through photosynthesis, so they give this carbon to the mycorrhizal fungi. And then they both benefit from this interaction. Before, we believe that mycorrhizal fungi were this, or the evolution of mycorrhizal associations were this important event in the evolution of plants that allowed plants to colonize terrestrial environments 450 million years ago. We have, let me, hold on, sorry. I don't know why this is not letting me. Okay, do you see my presentation now?
Yeah.
Did it change? Okay, we know this because some scientists, for example, in this paper in 2010, have found mycorrhizal fungi in the roots of the most basal, most ancient plants, which are liverworts. So we know that basically since the very beginning, plants were forming this mycorrhizal network, this mycorrhizal symbiosis, even before they have real roots, because liverworts, for example, only have rhizoids instead of real roots. So this interaction is very ancient. Even it has been found that plants had mutualistic fungi, also symbiotic fungi, living in their tissue before they even colonized terrestrial habitats. So
Adriana,
yes.
we are not seeing your presentation.
Okay. Yes, I suspected that was.
Yeah,
Oh,
we were on the title page and then you stopped sharing, and that was
okay. Yes. I don't know what happened.
Okay.
Sorry about
Oh,
that.
okay.
great. Thank you.
Okay. Yes. Thank you for letting me know. Okay, this is the one that I was telling you that even in these very ancient plants, they have mycorrhizal fungi in their rhizoids. And this was, I was telling you that even aquatic plants, before they colonize terrestrial environments, they have this mutualistic fungi that they interacted with. So this association, plant fungi, is really ancient. And we think it has been shown that after plants colonize terrestrial environments, and this colonization was facilitated by arbuscular mycorrhizal fungi, there was a very strong change in the composition of our atmosphere, right? The CO2 concentrations in the atmosphere was reduced dramatically, as you see in this purple arrow. So there is evidence that plants and their associated mycorrhizal fungi were crucial for basically changing climate on Earth and atmosphere composition. So let's look a little bit more about the microbiome of the plant roots. So I say the microbiome because these fungi have very similar functions that our gut microbiome. We have a lot of bacteria in our gut that helps us absorb nutrients from our food. So all these fungi that live in the roots of plants help plants absorb nutrients from their soil. So it has very similar functions. But in the roots of plants, they're not only mycorrhizal fungi, they are also root endophytes, they are plant pathogens, they are saprotrophic fungi, commensal endophytes, that are other fungi, fungal parasites. So there is a lot of diversity in the roots of plants. We're going to focus on mycorrhizal fungi, but I just wanted to know that there is way more diversity that is living in the roots of plants. And mycorrhizal fungi are a big part of the soil microbiome as well. We believe mycorrhizal fungi comprise about 30% of all the microbial biomass that is living in the soil, and they form these enormous networks that are microscopic that transport nutrients. And we believe they are basically the circulatory system of Earth because they are moving enormous amounts of nutrients, water, carbon, all over the soil and all over the world. And they have very important functions in the ecosystem and also for our and for the human use. For example, mycorrhizal fungi reduce the need for fertilization in crops, and basically they are the key for developing sustainable agriculture because they fix a lot of carbon on the soil in superficial and in deep layers of the soil. So they are really, really important for maintaining basically soil health and maintaining a lot of um, underground, basically nutrient flows. I wanted to show you this video that shows how important these mycorrhizal fungi are. I have this in YouTube, so give me 1 second. It's a very short video, but it's showing in real time how these mycorrhizal fungi are moving nutrients. So I hope you saw the speed of those networks moving all that carbon. And basically, these fungi are deciding at this speed where nutrients go, where they're going to trade with the plants. And it's like a very, very active network of exchanging basically goods between plants and fungi. And yeah, fungi have their own agenda and their own basically is like a brain, right? That is making all these decisions in a very fast pace. And we believe in this new article that came out like a couple of years ago that mycorrhizal networks move about 13 billion tons of carbon or CO2 or carbon all over the world. That's equivalent to more or less 30% of all the CO2 emissions that we produce. So mycorrhizal fungi are a very important carbon sink for ecosystems. So maintaining healthy mycorrhizal fungi are very important for our climate agenda. And let me speak a little bit about different types of mycorrhizal fungi because, for example, where Peggy was talking about my work, she said the word ectomycorrhizal fungi. So I have a couple of slides here to tell you more what is the difference. So there are like 5 different types of mycorrhizal fungi, but the 2 most important types of mycorrhizal fungi are ectomycorrhizal fungi and arbuscular mycorrhizal fungi. Ectomycorrhizal fungi are very common, for example, in upstate New York, because they form associations with many conifers and many temperate trees. They form mushrooms. So you probably have seen many mushrooms growing in the woods. Many of them are edible, and they form like a mantle of a layer in the outside of the roots. Those are ectomycorrhizal fungi. Arbuscular mycorrhizal fungi are more invisible. They don't form any type of visible structures, and they form associations mostly with herbs. In the prairie, for example, they are dominant, and in the tropical forests, they are also dominant. They form a lot of associations with tropical forests. And they are, arbuscular mycorrhizal associate to most of the plant species on Earth, more or less 80% of species. Ectomycorrhizal fungi associate only with 2%, but those 2% species account for 26% of all the plants on Earth, like all the coverage. So it's not that they are not important, because, for example, oaks and pines form ectomycorrhizal associations. Those are very important for many ecosystems. But for arbuscular, most of the crops that we grow form arbuscular mycorrhizal fungi, like corn or wheat or beans. All those form arbuscular mycorrhizal fungi. So they are both really important in their own way. Also, arbuscular mycorrhizal fungi are very old evolutionary. Those were the ones, for example, found in the roots of these, in mosses and liverworts. They have very few species globally, less than 1,000 species, and they specialize in phosphorus acquisition. Ectomycorrhizal fungi specialize in the acquisition of nitrogen. They are extremely diverse, more than 30,000 species globally, and they have evolved more recently. So I'm going to tell you a little bit now about what we do at SPON, because we think it's very important to, SPON is a nonprofit organization. And what we're doing is three things: map, protect, and harness mycorrhizal fungi. So with maps, we are creating these very high resolution global maps of mycorrhizal biodiversity to be able to identify, basically know what is where, everywhere in the world with very high resolution, and to identify, for example, hotspots or areas where conservation is important. I'm going to tell you more about that later on. And for this, we basically go to the field and collect soils. I'm going to tell you more about this story. We use statistical models to predict mycorrhizal diversity. And we also go back in the forests and on the ground to see if those predictions are working well. So we do a lot of field work. We use those maps and all the information that we gather to protect, to understand what are the functions that mycorrhizal fungi are doing on the ground, where are the important areas that we need to preserve, and also try to include those fungi and those important, basically fungal biodiversity in restoration plans or in sustainable agriculture, any type of conservation decisions. And we want to also harness the power of mycorrhizal fungi for example, fighting climate change, to develop sustainable agriculture, to do bioremediation, whatever they could be useful for, because it's very important that we use the power of mycorrhizal networks to solve basically environmental challenges that we have right now. So to map these mycorrhizal fungi, we go to on the ground, either in the woods, in pristine areas, or in agricultural areas, or in degraded areas. We collect soils, and we bring them to the lab. We sequence them. We use environmental sequencing to understand the mycorrhizal diversity of these samples. And then we have a list basically of species. We are able to know how many species are there, what species are there, if in many cases. And then we create these global databases. We didn't create this all by ourselves. We are collaborating with also Global Fungi and the Crowther Lab. There are also data agglomerators that have compilators, that have compiled thousands of samples from all over the world. And we are using this information for both ectomycorrhizal fungi in the top and arbuscular mycorrhizal fungi to create these global maps of mycorrhizal biodiversity. Also, the problem with global maps is that there are so many knowledge gaps. It's impossible to go everywhere in the world and to have samples. So we are prioritizing basically the places that are more underexplored, to go on expeditions and to collect soils and basically produce new data from these regions. For example, we were in Chile, in the Atacama Desert. We went in the Amazon in Ecuador, collecting with indigenous communities. We were in Kazakhstan. We have been in Mongolia, places where we have identified knowledge gaps. But of course, we cannot do everything by ourselves. So we have also a granting program that is called the Underground Explorers Program, when we grant money to local mycorrhizal researchers, but mostly from the Global South, so they can do their own research and also afterwards share those data with us so we can help create local expertise, local knowledge, and also they contribute to our cause of mapping the underground. If you want to know more about SPAWN, about this granting program, you can scan this QR code that is here to find out more about SPAWN's programs. And these are only 4 examples of great grantees that we have operating in the US, working, for example, in climate change, in reshaping the mycorrhizal communities in snow-covered forests in New Hampshire, or working on restoration in the tall grass prairies in Kansas, or in Texas, and also looking at microplastic pollution and how mycorrhizal fungi are affected by microplastic pollution in California. We have many, many more grantees, and also in the SPAWN website, you can see more projects from all over the world if you are interested. So what we do with this data is we're gathering more and more information every day. So we train our machine learning models with this soil biodiversity or mycorrhizal biodiversity data. We predict the mycorrhizal diversity at very fine scales, 1 kilometer square. We validate with also our independent data sets to make sure these models are working well. And we interpret this data to make sense of the data and also to make this information available for the local, for everyone, not only for other scientists, but we want to make sure everyone can understand and learns about the hotspots or all our findings, the importance of mycorrhizal fungi in ecosystems and all our research. So we develop tools to make this more easily accessible for the public, for decision makers, and from basically everyone that wants to use it. So this year we published a very important paper that we have been working on for a couple of years or more, about the global hotspots of mycorrhizal fungi biodiversity, and we found that they are poorly protected. So the whole message is in the title of the paper. So we took this data set of more than 30,000 points globally, and we apply our machine learning algorithms predicted mycorrhizal diversity for ecto and arbuscular mycorrhizal fungi. And we were able to do 2 things. First, identify the hotspots of mycorrhizal diversity all over the world. As you see here, there are many, many in many places. And And also we overlap those hotspots with conservation areas globally. And we found that less than 10% of those hotspots are being protected. So more than 90% are not protected. So that was an important result for us, because that gives us a north of what we need to do. We need to go identify these hotspots and basically advocate to create more conservation areas in areas where there is high mycorrhizal diversity, because all of the conservation areas in the world has been selected based only in plants and animals. Nobody have taken into account fungi. So for us, it's really important to start thinking about fungi when deciding where to create new conservation areas, for example. And yeah, we realized that actually mycorrhizal fungi are 3 times less protected than other organisms like plants and animals. So they're pretty much unprotected. We also, with these maps, wanted to look at uncertainty, because since we're working with models and with statistical models, we know we cannot be sure of everything that we're saying. So we want to be super transparent and identify these places where we really don't have a lot of certainty with the predictions of the model. So we can prioritize those areas to go back and collect more, because usually we have uncertainty because we have very little data. So we're trying to gather more data in these areas to improve our models for the next version of the data set. So with this data set, we also identified that montane grasslands are the most diverse ecosystems on Earth for arbuscular mycorrhizal fungi, and we found that conifer forests, both tropical and temperate forests, are the most diverse areas or ecosystems in the world for ectomycorrhizal fungi. And we also look at rarity, because for conservation, the number of species is very important, the richness, right, the hotspots. But we also wanted to identify what are the places where we have the more rare species, the endemic species, that could be very vulnerable also to deforestation or climate change. So we identify many places that have the rarest species in the world. Most of them, for arbuscular mycorrhizal fungi, for example, are in the tropics, and for ectomycorrhizal fungi are in the boreal regions. So those are also could be priority areas to protect these endemic species, for example. And we also found that the hotspots of arbuscular and ectomycorrhizal fungi are not in the same places. They overlap very little. So that's another important thing, that the ecology of these groups are very distinct, and they need their own conservation plans, basically. If you want to learn more about this tool that is called the Underground Atlas, if you want to explore these hotspots maps that we created, we developed this tool. You can scan the code and go look at your favorite area in the world. I have a little bit of zooming here in the Northeast, or basically in the East United States. As you can see, New York State has a lot of ectomycorrhizal diversity, is one of those bright hotspots. So we'll take a look and see how many species you have in your area. I also wanted to tell you a little bit about this other paper that we published this year, because it's the divergency between plant and mycorrhizal hotspots. Because since mycorrhizal fungi are associated to plants, many people tend to think that, okay, if I'm preserving as many plants as possible, then that's good for mycorrhizal fungi, so I don't have to think about mycorrhizal fungi, right? But we want to emphasize that this is not enough. Like, we need to think about mycorrhizal fungi as on its own, because these two hotspots don't really match. We found that sometimes there are a lot of plants and not necessarily there are as many mycorrhizal fungi. On other places, we found that in many places there are many mycorrhizal fungi and not as many plants. So there is a lot of mismatches, and actually there is not always, basically plants are not a very good proxy for understanding mycorrhizal diversity or mycorrhizal hotspots. Actually, this is the map for the overlap between plant hotspots and arbuscular mycorrhizal hotspots. And as you see, the pink areas, the fuchsia areas, are the places where they match. And you see, it's like you cannot even see it without zooming in. So they are very small areas where these two hotspots have a match. Same for ectomycorrhizal fungi. So we need to think about mycorrhizal fungi on its own instead of trying to use plants or animals as a proxy for the conservation of mycorrhizal fungi. And finally, I wanted to tell you about this other paper that we published where we analyze Earth's dark ectomycorrhizal fungi. So what are dark? What is those dark fungi? Dark taxa are taxa that are known only from a sequence of DNA, but they don't have any names. So basically, with the developing of DNA and sequencing technology, we have been able to understand better the diversity of fungi, or basically all taxa on Earth, but the taxonomy work that has to follow doesn't really match the speed of eDNA, basically. So there are many species that are known only from a sequence, but don't have a formal name. So we look at those and we realize that only basically that 88% of all the sequences that we're able to obtain based on environmental DNA, only 88% don't have a name. That means they are dark taxa. Why is this important? Why naming is so important? Because for conservation, a lot of the conservation is the species specific base, right? Species base. You preserve, I don't know, polar bears, right? And we have a scientific name for that species. And then you can channel a lot of efforts to preserve that species because you know what you are protecting. But when a species don't have a name, the protection becomes more complicated. For example, the IUCN red listing group only accepts species that have a name, right? And also that are macroscopic. You cannot protect microscopic fungi through red listing because you have to be able to identify entire populations, and you have to be able to show that these are breeding populations, etc. So there are a lot of requirements, and a lot of that comes from a name, from having a name. So understanding how much taxa are unknown, we can create research priorities to start naming those species and be able to actually put them, give them a name and maybe protect that species in specific. For example, at this moment, there are less than 1,000 fungal species in the red lists globally. And a lot of those are not even mycorrhizal fungi. So we have a lot to do in terms of species specific protection for mycorrhizal fungi. So based on this analysis, we were able to identify research priority metrics for all over the world. So we look at places that have a lot of species diversity, but also have a lot of species that don't have any names. And those are the bright hotspots here. So as you see, there is a lot in the tropics, but basically everywhere. where there are ectomycorrhizal species, there are hotspots of research priorities. So this will also give us a way to understand where to go next, where to focus our efforts. And then also is a very important call for supporting taxonomists, fungal taxonomists, because there are so few. It's a profession basically that is decreasing. A lot of people are not dedicating themselves to put names on things, because why? You can do it with DNA, right? But we're realizing no, putting names on these organisms is so important. So we're going from these global maps also in SPON. We don't want to stop this global view of what is going on. So we are working on very specific projects where we identify that we could make a difference and we can go deeper on the understanding of what is happening and hopefully try to influence some policy. So for example, we have been in Italy collecting in this very drought time, during a very strong drought, and we collected soils in place impacted by wildfire versus places that haven't been impacted by wildfire, to understand the effect of fire on fungal communities, to try to see what could be done there. We also have done a lot of, or we did an expedition in grasslands in Kazakhstan. Kazakhstan is an area that is going through a very rapid desertification rate due to overgrazing. It's a very dry area already. And after overgrazing, plants cannot recover. And then basically that starts turning into desert. So we're trying to understand what are the factors, what is the carry capacity, and what could be done in terms of management to reduce desertification and to reduce the impact of overgrazing on mycorrhizal communities. We also have look at, for example, the impact of soil erosion in Lesotho. Lesotho is a very interesting area because it has a lot of mountains. It's a very rainy area. And there is a lot of erosion because of the slope. So we wanted to also understand what is happening to mycorrhizal fungi under these conditions and what could be done to protect mycorrhizal fungi. We're also looking at the invasive coconut trees in Palmyra atoll. So coconut trees were introduced there a long time ago, and they have become an invasive species that is changing mycorrhizal community composition in these areas that are, in general, it's very pristine, but the introduction of this one species is changing everything because, for example, there are many species like Pisonia trees that are ectomycorrhizal, coconuts are arbuscular mycorrhizal, and then there is a lot of competition between those two groups of mycorrhizal fungi for space, for hosts. So we're trying to understand the effect of these mycorrhizal fungi on the effect of these invasive plant species in these trees. And there are many other examples. For example, we were in the Atacama Desert looking at what fungi are supporting agricultural systems in the driest desert in the world. And that was very interesting to see that actually there is a lot of mycorrhizal diversity in these ecosystems, and deserts are not really as empty as we think. They host a lot of diversity. So that was really interesting study. I just came back, for example, from Indonesia, where we were sampling inside national parks and outside national parks in impacted ecosystems to try to see what is the impact of deforestation in mycorrhizal fungal diversity. We also went to the Equatorial Amazon to look at the effect of indigenous agriculture in mycorrhizal diversity and how their traditional ways of food production are very sustainable and helping mycorrhizal diversity to persist over time. So we're trying to look at many areas and try to come up with scientific evidence that hopefully could be used to influence policy locally. So far, Spawn only was born 4 years ago, so we are kind of a young organization, but we're really trying to push hard to try to include mycorrhizal diversity in decision making, in conservation. In the 30 by 30 plants, for example, that have 30% protected areas by 2030, we really want to advocate for fungi, try to push for mycorrhizal fungi to be one of those criteria that are used for selecting this 30% of land that are going to be protected. So there is a lot of work there, but in the last Biodiversity Cup here in Colombia, we are collaborating with the Fungi Foundation, and then they were able to get some countries, the country level, to commit to include mycorrhizal or fungi in general for their conservation plans. So I think there's a lot of hope there, and we're working really hard to try to develop basically global strategies for including fungi in conservation agendas. And that's all I have to prepare, but I guess we can talk a little more in the Q&A section. I just wanted to thank the whole thank you for attending, and also the whole Spawn team. We're a big team with scientists, but also communicators. We have been featured in many media. So you are curious, we have a session on press in our website. You want to learn more about the different projects that we have been doing. We also thank the Global Fungi team that helped us compile this enormous global data set and many other collaborators that we have globally. Thank you so much.
Thank you. I learned a lot. I'd like to just start with a couple of questions and then open it up for everybody else. So can you talk about, I guess, two things. One is the threats to it. I mean, it's fascinating to learn that it's not exactly synonymous with the health of the plant community. So I'm a little bit baffled by that. Climate, desertification, what exactly? And then also, what what are some of the things that this is a group of activists that you're looking at? What kinds of things should we be advocating for? So both of those things. What are the threats, and what kinds of things, policies should we be advocating for?
Yeah, those are very good questions. So the threats, I think they're the obvious threats and the not so obvious threats. The obvious threats is, for example, deforestation, right? Because without plants, these fungi cannot survive. So of course, deforestation is a big issue. Soil pollution is a big issue. Basically, sealing of the soil is a big issue. But for example, the invisible threats are, for example, fertilization, right? Because plants are very happy when they are fertilized, but fungi are not. Mycorrhizal fungi don't like fertilization because when plants are fertilized, then they completely switch to basically obtain the nutrients on their own, and they stop investing in mycorrhizal fungi, and they basically become super dependent on these external inputs. So fertilization is a big one. Plowing is a big one too, because it disturbs mycorrhizal networks and it could reduce the amount of carbon stored in the soils. Basically not using cover crops over certain period of times, because the cover crops really keep mycorrhizal fungi alive. Fungicides and pesticides that we use a lot in agriculture. There has been some evidence that the fungicides that we use for pests, for fungi that are pathogens, also could be detrimental for mycorrhizal fungi. So basically, we are killing the friends that when we apply these fungicides and insecticides too, that could be, like, all those could have effects on mycorrhizal fungi. Basically, all disturbance will affect mycorrhizal fungi from one or another way, but I think those are the big ones, especially for agriculture.
What about extreme heat? Is that another one?
Yes, absolutely. Like fire would change the mycorrhizal communities, and depending on the intensity of the fire and the intensity on the area that is burned, then the recovery of the mycorrhizal communities will be slower or faster. It depends on many, let's say, local factors. But yes, the heat would affect mycorrhizal fungi. And of course, all the subtle, like the climate change factors, like too much drought, too much rain, all those also will affect mycorrhizal fungi. But we don't have a lot of information right now about the effect of those. We're just starting to learn, mostly in the long-term experiments, what could happen to mycorrhizal fungi in general. And in terms of policy, that's a good question, because I think for me, the most important thing that we should be advocating for is to really think about this fungi when decision makers are making conservation decisions about anything. For example, when you're doing restoration, think about on the underground as well. We can, if we think only about plants and bringing plants and the right plants, but those plants come with a substrate that is completely random. We don't even think that we also need to restore the native mycorrhizal networks of the native fungi that live in the soil. So thinking more, being more mindful about the way we introduce fungal and mycorrhizal species in management plans. Also thinking about specifically, be explicit about protecting fungi when thinking about creating new preserves, when thinking about basically all the conservation decisions at the tier F. There is the 3F initiative. I don't know if you guys have heard about it, but it's flora, fauna, and funga. We need to think about funga as well, the same way that we think about fauna and flora for all conservation. Yes.
So I'm understanding that native plants and native flora is the best thing to restore the fungal networks as well. Is that correct?
Yes, native plants, but also native mycorrhiza. Because, for example, there is a lot of mycorrhizal inoculum or mycorrhizal biofertilizers that are sold in stores that sometimes could have like exotic species that come from different regions and even different countries. So being mindful also about those, what comes in the roots of the plants already, that plants are inoculated with native species as well of mycorrhizal fungi. I think that's super important as well.
We have a question, and then you should all be able to unmute yourselves if you want to ask questions. We have a question in the chat. Have you done any research into the health effects of human contact with these networks? Is there science that supports the idea that connection with soil improves human immunity and health?
Oh, yes, that's a good question. I haven't done myself. I don't really study humans or the interactions with humans. I mostly work with plants and fungi, but I think a healthy soil makes healthy food, and healthy food makes healthy people. So I think there is a very direct connection that we really haven't understand
I think,
completely.
I think there's like, on the one hand, there, and Gail, I see you in 1 second. I think on the one hand, there's always concern about that the soil isn't sanitary, and for example, that it's not good for children to play in the dirt. And on the other hand, I also read the idea that it's good for children, even health-wise, even for, to play in the dirt. Do you have any sense about that or no?
Well, as a mom, I let my kid play in the dirt all the time. I think that's important. That diverse microbiome is gonna be good for him. So 100%.
I see we have two questions. Gail, two raised hands.
Yeah, thanks. This has been terrific. You mentioned plowing disrupts the fungal habitat. So is something like no-till agriculture a better thing because then it leaves? Okay. Yeah.
Yes, I think no-till agriculture would be ideal to preserve mycorrhizal networks, but also a lot of other important microorganisms in the soil and to preserve more carbon. So yes, 100% for that. Yes.
This has been an absolutely fantastic presentation. I learned so much. I'm totally fascinated by this now. So what your group is doing, what Spun is doing, is basically creating databases for the ectorhizomal fungi and the other kind. I forget what it's called. So ecto is more external, and the other one is underneath the ground, right? And the underneath the ground ones are the ones that are more the carbon sink, that suck up all that carbon and move it. They move it around underground. So is your research is basically starting from a point in this planet where it's totally polluted, from top to bottom. I don't know how deep the research goes, but how like are these diverse species disappearing as quickly as other things are because of the constant chronic state of pollution and the different environmental stresses our planet and pollution that our planet is under constantly? So is that like destroying them as we're speaking? And is the conservation planning pretty much the only thing that can help?
Oh yeah, that's a very good question. I want to thank you. I'm so excited that you feel inspired by this. Thank you for that. Both types of mycorrhizal fungi really move nutrients. One creates the fruiting bodies, the mushrooms, but also have very large networks underground, so they have very similar functions. Regarding your question specifically, I would say yes, they could be species disappearing as we speak. For example, I work with this endangered plant in Colombia. It's an oak species that is only found in Colombia in very small populations. And here in Colombia, we only have 6 host plant species that form ectomycorrhizal fungi. Everything else, like 20,000 more species, form arbuscular. So this one host is already endangered. It's being cut down at alarming rate. And I'm just so scared that basically there are many species that associate with this plant that I have found are like 700 mycorrhizal species. are disappearing. Yes, I feel like they could be disappearing. And this is only one case that we have been able to document
Mhm.
what is happening, because there are so many things that don't have names that we really don't even know. If they disappear, they are gone and we never even knew they existed.
And just
So that's one of the issues with this dark taxa, that we cannot quantify how much we are losing every day. But
Right,
yes,
so,
just
so
to editorialize a little bit, what you're saying is basically that these fungi are necessary to maintain the plant life.
yeah,
So when they disappear, we do have two other
thank
questions,
you.
Cassie. Okay.
Yeah, I'm gonna get
Okay.
off. Thanks.
Okay.
I
Okay,
mean,
Lori, and then I have one more question too. Lori Evans, you
Sorry.
need, okay.
Hi. Thank you so much. Very interesting. I had no idea of the different types of mycorrhizal fungi. So I have a two-part question. One, when we learn, like through the Bionutrient Food Association or Elaine Ingham's work on soil health, about no-till, about rock mineral dust and cover crops, is enriching the soil in that way, not with fertilizers. Does that promote mycorrhizal fungi growth?
yes, absolutely. Yes.
So is there something we can do when we plant that would increase? Are those the things we do? We focus on the soil, or is there something else to do? Like I've heard, and I don't know what it's called, there's a coating you can put on seeds before you plant them. But I think you were saying that some of them are native and some are not, which I never even knew to consider.
Yes, that's a very good question. Sorry, I was looking for an article that we have in our website. It's an explainer of how to have healthier mycorrhizal networks in your garden. So I will share that with you in a second. But yes, besides all those things that you mentioned, no-till, cover crops, rock mineral, I think having a little bit of plant diversity, for example, in your garden, that helps a lot. For example, having a row or native plants in between crops or something like that. I think those plants could really help, like islands of diversity for native species, native mycorrhizal species. Things like that could be beneficial. So
I just want to also, I'm sorry, did I interrupt you?
no.
Yesterday, I was at the Hudson Valley Nut Festival in Red Hook, New York, and it's amazing what this group, several people are doing to promote agroforestry, to bring back a hybrid chestnut, not a GMO chestnut, but also shagbark hickory nuts, and many others, to bring back what indigenous people used to have on this land. And they're using agroforestry with animals underneath, but they're taking very degraded areas. And it's amazing what you can do with soil when you give it the nourishment it needs, much faster than what we had been told as far as growing an inch of soil. Thank you very much to all of you.
We have a question from John, and then I have a question, and then maybe we should wrap up. John?
Thanks very much. Super interesting. I want to ask a question. As the permafrost melts, I guess the rumor is that that's going to release more carbon in the atmosphere. But as the ground there gets warmer, how does it impact any microbial fungal activity? Does it increase it and therefore sequester more carbon, or does it not make an impact?
Ooh, that's a super good question. I have no answer for that because there are a lot of different interactions. So maybe that could be beneficial for some species and not beneficial for other species. There are a lot of research about this in, I think in Sweden. In Sweden, they are, because basically as the permafrost melts and plant species start migrating north, basically because the conditions become more better for the plants, there is a lot of changes in ecosystem function and in plant species composition and mycorrhizal species composition. So it's very complex, and basically there are always winners and losers, and it's not super straightforward to say it, but basically these ecosystems are gonna be changed forever if they continue melting. Yes.
So I have one more question. And I know it's probably a complicated question, but how do the fungi make these decisions that they're making without any kind of central brain? How are they doing it? How do they communicate?
Yes, that's a good question. I think there are two hypotheses, basically. One is that they are trading based on what is becoming scarce in the soil, and then basically nutrients that are more difficult to acquire, then they will trade it for more carbon. And that's the trading hypothesis. There is another hypothesis that there is a sink and source. So they are bringing nutrients when they are abundant to when they are not abundant, and the other way around. And we don't really know exactly how they are making these decisions, but we do know that fungi are capable to making these split-second decisions of where to send nutrients and where not to send. We know also that fungi, in many cases, control the flow versus plants. Plants are more like at mercy of the fungi in this sense. But I don't really know a lot about fungal communication. And that's a little bit of a controversial topic right now because, for example, the mother tree hypothesis and the Internet of Plants have only been really actually been shown in a few systems. So there was like a publication saying that we are kind of overblowing this story, so we have to be a little bit more careful. But still, it has been demonstrated in a few systems and it's real. But it's better not to say that plants are talking to each other through the fungi, because the fungi have a lot to say there. They are not there only to transfer messages from one plant to another, because they also have their own agenda.
Okay. Well, this has been fascinating. I learned a lot. I think we all did. There's something awe-inspiring about it, I have to say. There really is. Thank you so much. I really appreciate it. I appreciate your work, and I'm definitely going to be reading more about this. This is fascinating. Thank you.
Yeah, thank you so much. And yeah, feel free to reach out. And yeah, I'm glad to hear you learn from this.
Oh, I think we all did. I think we all did.